we should start. While before the coffee break, we mainly talked about hazard identification and causality. Now we are after the coffee break, that's for sure. And we will focus on hazard characterization and dose response modeling. So there will be 3 speakers, while Marc Aerts, the first speaker, will focus on the introduction to dose response modeling and the current practices and challenges around it, while then the second speakers will talk about combining the evidence in dose response modeling using multivariate models, and the third speaker then on other approaches to combine evidence. Marc Aerts is a professor of biostatistics and director of the Center for Statistics at Hasselt University in Belgium. He published methodological work on mathematical and statistical models for infectious diseases, microbial risk assessment, and food safety, dose response modeling, toxicological and teratology experiments in various statistical approaches. He serves as associate editor for biometrics and statistical modeling, and he is chairman of the Masters of Statistics program and teaches courses on modeling infectious diseases, computer-intensive methods, data mining, categorical data modeling, and microbial risk assessment. Marc, please.
Thank you, Mr. Chairman. Thank you all, also organizers, for giving me the opportunity to collaborate to this very interesting event. When I was thinking about how to fill in my time slot, you have to think about the approach you are going to take. And so my approach was to think back in some sense about think about the existing guidances which are available, EFSA and EPA. I was a bit involved in the update of the EFSA guidance, and I was then thinking about, okay, I suppose EFSA will regularly update its guidances, not next month, I suppose, but I was thinking a next update should go more in the direction of guidance for multiple endpoints and multiple studies. And from that point of view, I thought, okay, let's look at the current status of that guidance, and let me present a series of possibilities to, from a methodological point of view, to look at possible approaches to deal with multiple endpoints and multiple studies, and of course, then we meet multiple challenges. You might be frightened by 48 slides, that's just to frighten you. But if you have a copy of the slides, you will see that about 15 at the end are what I did. I had to do the exercise myself. I went through the 2 documents of the guidances and copy-pasted all sentences, literally, which were mentioning multiple endpoints and multiple studies. So that's at your availability already. So subtract -15 in the number of slides. Okay, let me start with just setting the scene quickly with 2 examples, and the next 2 presenters will go into these examples in more depth. So this is a single study situation with multiple endpoints. So a toxicity study with rhodocilsulate in rats. And just to show some data and dose response and BMD, so benchmark dose estimations, I show 2 figures which are taken, if I remember well, from the paper from Wout et al. And Wout, the next presenter, is going to talk much more about it. So I will not say too much about it, Wout. But just to point at some issues we have to think about, multiple endpoints. So we have to think about how are we going to combine that information. We expect that they are highly correlated, so do we need all of them? Should we group them? They have their own scale, probably it's hard to see, but the maximum response is not the same. Do we have to think about that and issues like that? The second example is an example where Matt will go into in much more detail. It's a 2-study example with 2 endpoints. So it's a long-term toxicity study where rats were exposed for concentrations of 3 MCDP, PD, and we have 2 endpoints, kidney tubular cell hyperplasia and nephropathy, which now are quantile or binary, whereas I think the others in the first example are all on a continuous scale. So that's another thing which plays a role, of course. And just to show you a bit about the data, I think these are the male nephropathy data from one of the studies. And what you see is
a dose-response fit of one of the models, because you have another multiplicity, namely being you having multiple models. And the approach taken here is that you show the dose-response model is chosen of that endpoint with the lowest BMDL. So just also to have some notation, although I'll keep maths as simple as possible, no maths, essentially trying to focus on concepts, but in a single endpoint, single study, we have concentration, doses, and endpoint observations, and sample size. They can be binary, quantile, or continuous. In a single study, we typically have, of course, multiple endpoints, M endpoints. They can be of a mixed nature. And then, of course, really getting into apples and oranges, as we have seen before the break, with many issues of heterogeneity in case we have multiple studies, even with a single endpoint, but especially, of course, when we have multiple endpoints, which can be varying across the different studies in number even, and so forth. Another important issue is, of course, that often we only have aggregated data. So when looking at multivariate structure, ideally we have the raw individual data. I'm not going to talk about hierarchical structures like data on fetuses within a litter, cluster type of data, but that's another layer of complexity which can be accommodated. Okay, so the next step is to look at the current guidance for single endpoint, and before going to that, I just want to have like 4 steps, obvious steps, but we'll come back to that when we go to multiple endpoints to look at simple pragmatic options to work with. So we have, of course, the layer of the data. We have the intermediate step where we fit a dose-response model, several potential models. So there is a multiplicity factor 2. That leads to corresponding to a BMR, a particular BMD, a benchmark dose estimation, and then we go to our confidence interval and the lower limit. So remember these 4 steps. We'll go back to that. This is just an example for continuous data, the 4-parameter Hill model, and the parameters have a particular interpretation, and this is the dose-response model, the BMR, which is here 5%, and increase in risk, and here we have the BM and the BMDL. Okay, so let's turn to the guidance at this point in time regarding single endpoint, single study. It's an update from about a year ago, and so the recommendation now is clearly going into take dose response modeling, go to the BMD. We have multiple models, as you can see in the guidance. In both cases, even in quantile situation, we have more models at our disposal than in a continuous case. How do we deal with the multiple models? Well, we use information criteria to measure goodness of fit and to go to a model averaging approach. So that's a way where we now deal with the multiplicity of the modeling in some sense. So model averaging is recommended for determining the BMD, the BMDL, and the BMDU. It provides a simple but helpful, I think, flowchart, and it's also recommended to not only report the lower limit but also the upper limit to have a complete review on the accuracy or inaccuracy. I'm not going through this flowchart. I'm just pointing at the central part here where it says model averaging software. Is it available? Of course, here I just want to point out the fact that the availability of software is an issue too. Often software is available, but as a package in R or whatever, and not really user-friendly for the type of application you have in mind. So if it's available, you can do model averaging. That's the idea, the recommendation. If not, we have multiple models, and we look at all models which are essentially fitting equally well with a kind of simple criterion, and then report confidence intervals for all selected models. But in the end, we have to suggest a point of departure, reference point, and then it says, well, use the lowest BMDL and the largest BMDU. So that's the guidance for a single endpoint. What about multiple endpoints and multiple studies? In the update for EFSA guidance, it's pretty limited. So the current guidance says, well, if you have multiple endpoints, select that endpoint resulting in the lowest BMDL. Of course, other additional information can be taken into account to have a pre-selection or to take it into account while selecting the endpoint. Case by case, so in the end, general principles but very limited practical guidance regarding multiple studies. Well, the use of covariates is suggested to account for heterogeneity, like fixed or random effects, to take that into account. I also had a quick look at the EPA technical guidance of 2012. Again, the details are in the last 15 slides. It reports more about specialized models with references for multiple outcomes and multiple studies. It also advocates the use of Bayesian approaches, that the risk assessor, based on the human exposure situation, should perhaps select studies first, and also endpoints could be selected first as representative to the effects in a particular target organ. Of course, simple approach would be combining data sets, but as we have learned, and everybody knows, be careful, the heterogeneity is there, and it necessitates more complex modeling. This last one refers to the fact, well, there are multiple models. If you have to apply multiple models for multiple endpoints, for multiple studies, and try out all combinations, it's like a job for, not for a week, but for the rest of your life. So you might have or need to limit the number of models from in the start. But again, the point I want to make is, it does mention more specialized methods as compared to the EFSA guidance, but in general, but it remains very general in principles, and the practical guidance is limited. So that was my point of view, to go from there and to think about pragmatic, simple approaches, starting with the current practice, and think about methodological ways to get further. Is it worthwhile to extend to multiple endpoints and studies? Obviously, I think yes. What's the price to pay? How far to go? So the question is, how detailed should the model be? Can we think about a common BMD, so a common BMD across different endpoints or studies, things like that, are popping up, and so forth. So let me quickly go to multiple endpoint approaches. So this is a type of data we are looking at. I'm starting with simple approaches, and therefore I go back to the 4 steps. So you could really think about simplification at the real end, at the BMDL end, or step 1, take one step back at the BMD, the benchmark dose itself, or fully at the beginning, the data, you could simplify it over there, or somewhere in between, in the middle, could you combine and simplify it by the dose response model. So let me start with going to the real endpoint, so to say, the BMDL. And this is, as I understand the current practice, you have separate analysis for each endpoint, and you can follow perfectly the current guidance, model averaging, and so forth. And in the end, you take, from the perspective of protecting against the most sensitive outcome, the minimum of those BMDLs. Of course, there could again be some pre-selection be involved. I mentioned here 2 versions. Now I'm talking about the univariate ones, which refers to the fact that you do separate analysis for each of the endpoints as if you have only one endpoint. Of course, this is very easy, but of course there's an obvious concern about, these have different accuracies. Of course, you can in the end report also the maximum BMDU, but you're getting in, you have a concern about, are we not being overly protective or too conservative? Because the more endpoints I have, the lower the minimum will be, obviously. Another issue is there are statistical flaws. Taking a minimum of lower limits of a confidence interval, what are you having at that point in time? Is it still a limit of a confidence interval, a lower limit? I don't think so as such. It's not defined in that way. One step back would be, okay, let's go one step back. Let's go like to the point estimate. That's our BMD. We have M of them, same philosophy, separate analysis, but at that point we take the minimum. That's still like the most sensitive outcome. So it's very close to what we had before. Essentially the same is used. The only thing is that if we can work with the inference of a minimum of estimators, and we have inference tools, then we can really determine the BMDL as a lower bound of a confidence interval and the BMDU as an upper bound of a confidence interval. The disadvantage is a bit that the inference might not be straightforward, but there you could rely on simulation methods like the bootstrap. At least in principle. I'm not saying it's always straightforward to do so. So, let's go to the really starting point. Then we have our data themselves, and we have M outcomes and endpoints. So one way would be to combine these and simplify matters from in the start rather than at the end. One way to do it, they might be, of course, of different nature. So one way to do it could be, okay, let's all we transform them in some sense to an adverse event. So if it's a continuous event, let's use a threshold to have an adverse event. And then we have, let's say, binary endpoints, all of them. And we can say, okay, let's define a single endpoint as the event, as an adverse event, which reflects at least one of them is adverse. So that's also, from a most sensitive or protective point of view, a logical way of working. And then you have one endpoint, and you can do what you are used to do for a single endpoint. So in that sense, it's also very easy, so to say. There's also, again, the concern, of course, the more endpoints you have, the lower the minimum will be. Is this what you want to be? Perhaps. I mean, it's not only about statistical issues, it's also that's from a toxicological point of view, or a general point of view, what you aim at. Do you really want to be very protective? Yes, I think so. So it's not clear which way to go, I think. Of course, instead of making this kind of simplification, actually you can write this as a product too, if it's indicators. You can turn into another type of product, a weighted average on a weighted geometric mean. And that's what Coffey et al. Have suggested. So you could take the original endpoints, but they recommend or suggest is to use what's called the desirability function, which can be defined for any type of outcome, binary or continuous. It scales from 0 to 1. 1 is like the value which is reflecting what you would expect for a completely healthy animal, and 0 would be very unlikely for a healthy animal, and you could discretize it too. So this could be a step function, this desirability function. This is a bit a subjective choice. The weights too, but on the other hand, it gives you the possibility to think about certain endpoints to be more relevant than others. So that's the strength and in some sense also the weakness or the subjectivity, but that's something which plays anyway. We have to add information to the data because data is so limited. Anyway, the point is again that you end up with a single endpoint, and from there you go. And you can follow the current guidelines. Okay, you could also try to intervene in the middle. So you could, look at all endpoints together and fit like one model to them. Does that make sense? Well, you can do that, and you can include the endpoint label, let's say, indicator as a covariate. Or you could have, in other words, endpoint-specific parameters. And so if all parameters are different, it's like a separate analysis, but you use one model. It's not that you use for one endpoint a different model than for the others, so it's one model to all endpoints. But if you take a very flexible model, I just took an example here, again the 4-parameter Hill model, where I only took the first parameter, which is the background response, to be endpoint specific. In such a situation, the BMD will be common to all endpoints. Of course, if the potency or the steepness parameter would differ from endpoint to endpoint, the benchmark dose would also start differing from endpoint to endpoint. But then, well, perhaps this makes sense as a framework to test for a common BMD. Does it make sense or not? That's my timer. I don't know why. I still have some time, right? Okay. So that's the idea here at the level of the response model. Okay, let's then turn, this was all like trying to simplify and fall back to, as far as possible, the current guidance for a single endpoint. This is a starting point, the full multi-dimensional endpoints. Yes, and of course, in this way, you, because the others might only partly take the correlation structure into account, and if these are continuous, you can think of a multivariate log normal distribution or something like that. There are other approaches, the use of copulas to combine the marginal distributions. This is a statistical word on its own. And with this multiple model, like you model all at once in a multivariate way, you could go back to all methods we have described before, which I presented in a univariate way. But of course, it gets more complicated to think, and you have so many choices to make. But of course, it allows more than in the previous case to have different dose response model for the different endpoints. And, but again, it generates so many possible models you can apply that this is certainly a challenge. And of course, you need individual data because you're looking at the multivariate individual observation of interest here. But suppose you could do that, an obvious way, you know that you expect this to be highly correlated. So like in the first example, 13-dimensional, if I remember well, perhaps you can reduce this to 2 dimensions and have only a loss of 10% of information. So that would be an obvious thing to think about. I'm not saying that this is the right way to go, but at least you can think about it. And so you end up with only 2 principal components or extensions of that ID. And actually you could see what these principal components are meaning in a data-driven way. Perhaps there's something like a biological effect they represent. I don't know, I'm just guessing, so to say, or thinking about it. And then you have a limited type of model, only bivariate, which is easier, so to say, than a 13-dimensional one. But again, you need individual data. That's a limitation. The next one is just an example to think about, in the 2-dimensional space, a kind of adverse event which you calibrate in the control group, and that increases, of course, in this example, when you have active doses, really not by endpoint, but in a multivariate way. Now, this could be 2 original endpoints, or this could be like our 2 principal components from the previous exercise. And then you turn it into adverse events, and what this method suggests, okay, choose these 2 thresholds so that this is a 5% probability area for the control group. Of course, this has no unique solution, except if I add a constraint, like on the standardized scale, I take these 2 thresholds the same. It's just an ID, an option. And from there on, you define this increasing probability of an adverse risk, and you look at the extra risk, and you go further as you've done before. So there are other approaches existing, and this is my final slide for this part of the talk. Structural equation models, I feel also that's an interesting approach. The idea there is that you think about all these observed endpoints as manifestations of one or more underlying latent variables. So perhaps also there are groups of endpoints, and you could have 2 latent variables, or you could just have the data speak for itself. And then you turn from there on the scale of that latent scale, or also a kind of latent adverse event. That might be something to struggle with, because that's on a latent scale. What does this mean? But it feels like it's going also into direction to some common process which I am not fully understanding. I'm not modeling it yet, but it's like leading to what the endpoints show me. So that's like a statistical approach of some common process. But of course, you can really start modeling mechanistic processes. And here I just mentioned two approaches I came across by Choi et al. And Limscomb et al. This is a mechanistic model. This is a step further, PBPK model, where from the mode of action of the chemical, they go to a mechanistic model and then to a kind of empirical model and apply it and determine the benchmark dose. So, quickly, summary, many approaches. We have to think, I think, again, I try to think about update of the guidance. It's not obvious which way to go, but at least I see options, pragmatic ones, which could be implemented quite easily. I'm not sure whether everybody's convinced that this would be the right way to go, but it's not only statistical arguments. Many things play a role, of course, aggregate versus individual data. Suppose you have, that's for the multiple studies, a mixture of that, also that availability of software. Okay, let me turn to multiple studies. I'm going to be very short there, because so many things have been said in the presentations, very nice presentations. I enjoyed them very much before the break, and I think everything there concerns and bias adjusted and whatever applies here too. In this kind of setting, there's even, I think, so limited number of data aggregated, so many limitations. On the other hand, we need a reference point. We need to go further. We need a value to work with. This has been discussed and said. Obviously, in the end, you can apply standard meta-analysis approaches. I'm just referring to an EFSA report in that direction with cadmium. I just want to mention 3 existing things as, again, something which could be applied. And the EPA one uses the CAT-REG approach. CAT-REG refers to categorical regression, and it allows to do a meta-analysis for multiple toxicity studies, at least if you convert all endpoints in an ordinal categorical way. So that's existing methodology. There is also, coming back to model averaging as it's in the current guidelines, there is available methodology and software to look at the Bayesian model averaging for random and fixed effects meta-analysis. There are things developed like network models as a kind of intermediate step to PBPK models, which looked at similar or topics or benchmark dose estimation. So I think these could be useful things. But again, meta-analysis is, of course, tricky business. So, conclusions, multiple challenges. Well, I put them here in a general way. Could we have a consensus of an acceptable starting point? As again, from the point of view of guidance document, because there is now a kind of vacuum, so to say, when it goes to multiple endpoints and multiple studies. That's at least my perception. So I think there could be a need to get more guidance, that people have more guidelines to how to work. But of course, while keeping consistent with the current guidelines and making optimal use of data structures. So we have concretized this in some questions which are on table in our discussion group in the afternoon. So thank you, Mr. Chairman. Thank you.
Thank you very much, Professor Aerts. We keep in mind the cry for more guidance in this area also.
Well, that was my perception.
We have time for one burning question that cannot wait till the afternoon breakout session. Nothing burning at this point.
Okay.
So we continue then with our second speaker, who will talk about combining evidence on multiple endpoints and dose response assessments, multivariate models, Professor Slob. Professor Slob studied biology at the University of Amsterdam with theoretical biology as his main subject. This is where he obtained his PhD in 1987 with work on applying statistics in biological research, including log transformation and power analysis. Wout Slob worked at the RIVM from 1986 to 1995 as a consulting statistician, and during that period he became gradually more interested in toxicology and risk assessment. From 1996 on, he specialized in methods for risk assessment on substances, including dose response modeling, exposure modeling, probabilistic risk analysis, and toxicokinetic modeling. As an expert in these quantitative methods, he played an important role in many international committees. Wout Slob developed a software program for analyzing those response data, PROAST, which is used a lot nationally and internationally in the context of risk assessment, as well as in the context of scientific research in general. From 2000 until 2010, Wout Slob was also appointed as a professor for quantitative risk assessment at the Institute of Risk Assessment Sciences, EDAS, of the Utrecht University. Professor Slob, please.
Thank you. That was a quite adequate introduction. I was asked by the organizers of this workshop to talk about multiple endpoints in dose response assessments. And I will actually do that. But not for the whole talk, so I will also address a couple of other issues at the end of my talk. I will notice you about when this happens. I will not talk very much about multivariate models, but Jose did not allow me to change the title, so it's still there. Okay, multiple endpoints. Well, we can have different endpoints within the same study or among different studies. And when we derive point of departures, which might be BMDLs or maybe NOALs, then we often see that those values differ. And then the usual conclusion is the endpoint with the lowest point of departure is the most sensitive endpoint. Right? Any objections? No. Oh, wait, we are going too fast now. So, I'm going to challenge this conclusion, even though there were no objections. First thing that we need to keep in mind is that the point of departure itself is an uncertain value. So when we have an NOAL, then different NOALs might be different simply because of imprecision. So, for instance, when you think about a study with 5 animals and another study with 50 animals, then the NOAL in the second study might be lower simply because of higher statistical power. So statistical sensitivity is not the same as biological sensitivity. There's the non-equal sign. When we are dealing with BMDLs, then those BMDLs may also differ due to imprecision. And the nice thing about the BMD approach is that you can make this visible. So let's take this simple example. We have two endpoints A and B, and these horizontal lines are the confidence intervals for the BMD. Then this means actually that the true BMD is somewhere between, let me see, where is the... I did not practice. Which button is the light? This one?
I think you have to turn it.
Oh, I have to turn it. And yeah, there it is. So the BMD, this says this is a confidence interval, so the BMD could be somewhere in that interval, but we don't know exactly where. And the same for the other endpoints. So we cannot really make a statement that the BMDs might be the same, or this one might be higher than that one, or this one might be lower than that one. We simply cannot tell because of the uncertainty. So the fact that the lower bound of the confidence interval, the BMDL here, is higher than this one, does not mean that it is less sensitive. We simply don't know. When we look at endpoint C, then we could say, okay, well, at least we now know that endpoint C is more sensitive than endpoint A. At least that is the evidence we have here. But still, we cannot say if endpoint C is more sensitive than endpoint B. So you cannot draw conclusions based on single numbers. That is something that experimental scientists often forget. If you want to prioritize, you need a complete imprecision interval and not just one single number. Good. Let's have a look at an example. This is a subchronic study. There were, as you probably know, they measure subchronic studies address a lot of different endpoints. And here I applied the BMD approach and calculated those intervals for all the endpoints, showing a significant trend. And then, indeed, as you can see here, those intervals are maybe quite different. By the way, the pro software calculates 2 confidence intervals for each individual endpoint, which you can see here. And that is because the guidance says that you should normally use 2 models, the exponential and the Hill model. And in this case, as you can see, the 2 models perform quite similarly. They actually, well, they more or less say the same thing. But the endpoints differ. And one thing I wanted to make clear is that, oops, this can be done, this type of analysis can be done in one single automated run in the Pro software. So it is not a lot of work. It may take you 10 minutes. You have to wait a little while, drink a coffee, and then you have the results. And then you see in this case that the different endpoints apparently are not all equally sensitive, because some of the intervals do not overlap to each other. However, this relates to BMDs that assume that the benchmark response is 5% for all the endpoints. So, and that means a 5% increase in the average level compared to the average level in the background, in the control group. And then you might wonder if, for instance, in the neutrophiles, the lower endpoint here might be lower because of the fact that 5% does not really compare to, let's say, 5% in cholesterol or maybe red blood cells or whatever endpoint. So maybe a 5% change is not really plausible to consider that as equivalent for different endpoints. That is sort of an intuitive notion that I think most of you will have. Okay, so if we think about that, so this is the crucial question: do we really, can we really use the same benchmark response for all those different endpoints? Well, there has been quite some work that was done on that. People were questioned, so sort of an expert elicitation. What do we think should be the appropriate benchmark response for all those different endpoints? And toxicologists were not really able to answer that question. And then, more or less coincidental, I developed a theory some years ago, which I call the effect size theory, which, by the way, is valid for experimental data in general, at least continuous data. And this theory might bring us a little bit further. This theory says, based on very fundamental principles, that it is plausible to scale the benchmark response to maximum response, in particular by the logarithm of the maximum response, where the maximum response is expressed as a fold change. I don't have the time to go into details of the theory, but just give you a very brief summary. This theory also predicts that the logarithm of the maximum response will be equal to a constant times S, and S is the within group standard deviation on the log scale for the log responses. So this is what the theory predicts. So what did I do? I collected as many data-rich endpoints, and for each endpoint I estimated the value of M and of S. And as you can see, these are the experimental results, or the empirical results. The prediction is confirmed by these data points. It's quite adequate. And because of this correlation, we could also say, in those cases where we don't really have a lot of information on the maximum response, because that will happen a lot in practice, we could also use the within group standard deviation as a proxy, as a surrogate. And okay, well, this is very briefly the theory, and then I thought, wait, hang on, so maybe those differences in endpoints that we saw before, or that we usually see with a fixed benchmark response, maybe that disappears when we use these scaled benchmark responses. So, before I will address that, just a small intermediate remark. This is the empirical relationship with the fitted prediction from the model, and as you can see, there are some red triangles, and those triangles are the endpoints for which I had a lot of different studies available so that I could estimate an average value for the maximum response and the standard deviation. The other black circles are individual dose response data sets, and they are, of course, very imprecise. Therefore, they are much more scattered. So this gives some additional confidence in this relationship. Anyway, when I go back to the first example where we had used the same benchmark response for all the endpoints, 5%, I thought, okay, let's now see what happens if we derive this endpoint-specific benchmark responses, according to the theory I just discussed. And now we see that those intervals are much closer together. Actually, you might hypothesize that they actually do have exactly the same benchmark dose, and the remaining scatter is just uncertainty in the data. Here's another example, the famous study on rhodocylsilane. The study director is in this room. Do you want to know his name? That is Ruud Woutersen. This was a very useful study. I used it for various purposes. Here again we see those intervals, BMD intervals, for a fixed 5% for all the endpoints, and then if we use the scale benchmark response, we can again see that those intervals are getting much closer together. And again, you might hypothesize that in reality there is only one BMD for all the endpoints. I have some other examples. I'm trying to gather studies that report clear effects in multiple endpoints. So if you have suggestions for studies that I can use to further validate this theory, that would be, of course, very useful. Okay, you might argue now that there is some remaining differences in confidence intervals, so they do not completely overlap always. However, here I use the incidentals within group standard deviation of this particular study, which might be a pretty bad estimate of what we might call the true value of that standard deviation. And this might explain the remaining small differences that we still have here. Anyway, I thought, well, okay, when this theory were true, so if it were really true that the one study is just, when all the endpoints are actually equally sensitive and have one BMD, then maybe we can model that with a model that reflects this theory by replacing the maximum response by QS, which is the prediction of the theory, and then see what happens. So we can fit a model where endpoint is a covariate, which means that we have different values for the standard deviation, depends on the endpoint. Of course, we have different background responses for depending on the endpoint. They clearly are different. And then we have just one common parameter Q, which is in that relationship, one common steepness parameter, and just one common BMD. Let's fit this model to those data. And this is the model fitted to the rhodocylsilane study. And what you can see that this appears to give a pretty good fit. And actually here, there's only one single benchmark dose that you might estimate from all those endpoints together. And of course, this raises the question, how do we now establish a benchmark dose confidence interval for the whole study, for this single BMD? And then the problem is that we now are faced with the fact that these observations are no longer independent because they are measured in the same animals. So we will have correlations among the residuals of the curves, and so the challenge now is how to do that. And here we have the multivariate methods that I had in the title. I would suggest that this should be possible using multivariate models, or methods, which I did not yet work out, but that can be done probably relatively straightforward. This is just to show you the fit of the model with the separate endpoints, which gives a better view of the goodness of the fit. And this looks quite convincing, I would say, maybe with a small exception here for the neutrophiles, but there might be particular reasons for that. So this model reflects that all endpoints are equally sensitive, and we get a pretty good fit. So the hypothesis, at least, is not rejected. I'm not saying that it's confirmed, by the way, but I'm saying it's not rejected. And this is just an illustration of the correlation between the different endpoints. You can calculate those. And that is needed to calculate the confidence interval, but it might also be interesting for biological reasons. So, for instance, ALT and AZ are clearly correlated. It would not surprise you that red blood cells is highly correlated with hemoglobin. and also with these parameters MCHC and MCV. So it's just to illustrate this. Okay, so the conclusion might be that in the various studies examined so far, endpoints seem to be equally sensitive, or at least close to that. And that might be an interesting conclusion, because if that is the case, we could just calculate one single benchmark dose for the whole study using multivariate methods. Okay, this is the end of my talking about endpoints within studies, and I now want to address a couple of other issues that I think are important for the discussion in our group. So this is a very simple example where we have 4 different studies, 2 rat studies and 2 mouse studies, and 2 subchronic studies and 2 developmental studies. And currently the risk assessors would conclude probably that fetal body weight in the developmental study is more sensitive than the subchronic endpoint, and mice are more sensitive than rats. And again, my question is that correct? Now, when we look at PODs and ask the question, why do those values differ? Well, the obvious explanations and the usual explanations are, well, we are talking about different endpoints, we're talking about different species, exposure durations, etc. Those are the factors that risk assessors normally have in mind, but of course, it might also be that the studies differed in strains, they were done in different labs, they use different diets, the study conditions were different, and so on. And this might be, in reality, the reason that those NOALs or BMDs were different. And then, of course, as I said earlier, there's uncertainty in the point of departure itself, and I would address all those factors together as, summarize them as study replication error. In other words, when you do the same study where those first factors are the same, you will get different NOALs as well. So, let's see how that works. I give you a couple of examples from the literature. Oh, this is going too fast. First of all, let's try an example where we compare endpoints in distinct studies. In particular, Ganer et al. In 2007 compared NOALs in a 2-generation study versus subchronic studies. And what they saw here is that this is the 2-gen and this is the subchronic NOAL. What they saw is that they are very nicely correlated. And this line, the blue line, is the identity line. So on average, the NOALs in both studies are actually the same. And now you might say, well, for some compounds, the subchronic study is more sensitive, and for other compounds, the 2-gen study is more sensitive. However, what Ganer did is she also looked at the variation between replicated studies. So these are in the lower plot you see the lowest versus the highest NOAL in the replicated subchronic studies, and then you see that they differ almost by the same magnitude as we see between the studies. So the variability within the same study is very close, or it's very similar to the variation between the studies. So this indicates that in reality, maybe all that scatter is just replication error. It doesn't relate to real differences. So, conclusion, it might be that stability replication explains the observed differences between 2-gen NOALs and subchronic NOALs. And that there, in reality, you might hypothesize there are no differences. Okay, next example is on species. I will talk a little bit about species because it was already addressed various times today. This is a list of studies that have found that on average, species are equally sensitive. The first one is on rabbit versus rabbit, rat versus rabbit. The second is on rat versus mice. The next one is also rats versus rabbits, and the last one has even, let me see, 6 species, including humans. And again, we see that after allometric scaling, the kinetics, this is on kinetics parameters in pharmaceutical studies, actually are equally sensitive to chemicals, on average. Okay, then let's have a closer look to this one of those studies. Here again, we see the NOELs of rats versus rabbits, showing this similar scatter as before. And again, the identity line illustrates that on average, rats and rabbits are actually equally sensitive. Now, does this also hold for individual compounds? That is, of course, another question. And when we look at study replication error, we again see that this scatter is of the same order of magnitude. This seems to indicate that, again, this scatter is just study replication error, and that even for individual compounds, it might be that rats and rabbits are actually equally sensitive. And in a recent, much larger database, we have done this analysis again, and with somewhat more robust statistical methods, I found, again, a very clear indication that actually even for individual compounds, there are no interspecies differences in NOELs. So it might be that those interspecies differences that we are talking about very often might not be as large as we always thought, and it might even be that they are, in some cases, absent. They don't exist. Of course, this is again a hypothesis, and this was only a couple of studies, and we would need more research to find out if this is a theory or a hypothesis that stands. So, my main message, or my more general message of my talk is that before addressing the question what to do with multiple dose-response datasets or multiple point of departures, we need to know what those differences come from. And we cannot just directly conclude if we have two PODs that we know where they come from, because it might be different labs, data errors, or whatever. So let's have a couple of hypotheses. First hypothesis would be all endpoints within a given study are more or less equally sensitive. So we can use the whole study for estimating one single BMD confidence interval. Another hypothesis would be that interspecies differences are in reality minor. And again, when we have studies using different species, we can maybe also use them for estimating the common BMD. And a third hypothesis, I know people are working on that, comparing the impact of exposure duration on the point of departure. And clearly, exposure duration does have an effect on average over chemicals. But there are some indications, I think, that again, the remaining scatter is explained by replication errors, so that we may just only need one constant factor to correct for study duration, or exposure duration would be better. And similarly, of course, we can think of other hypotheses that we may investigate using data where we have replicated studies for the same factor. Now, if these hypotheses are approximately true, we can handle the problem of multiple studies by simply taking the geometric mean of the study BMDs and calculate the confidence interval for that mean value. And we might think about taking the benchmark, those confidence intervals, into account for as weights for the calculating a weighted mean value. And in the case of different exposure durations, we would at least need a constant to correct for that exposure duration, of course. Okay, and well, what happens is then, of course, when you have more study, you are rewarded in the sense of getting a higher, lower confidence interval. So the fewer studies you have, your lower confidence interval, well, your interval will be wider and the lower bound will be lower. Finally, a very simple numerical example. Suppose we have 4 studies with these 4 PODs, just to give some feeling of what happens. There's no difference in exposure duration here, so we just take the mean, the geometric mean, of course, which is 100 milligram per kilogram in this case. And the lower confidence interval bound happens to be 19, I should say, yeah. And which is very close to the lowest value. Now you might say, okay, then we just take the lowest value because there's no difference, but of course, And oh yeah, then we could use this as the POD. But of course, when you have more studies, the lower bound will be higher, and when you have fewer studies, the lower bound will be lower. So you will have a more precise methodology to give a point of departure for this compound in the case of multiple studies. Thank you very much.
Thank you very much, Professor Slob. It looks like that it might be simpler than we thought in the beginning, so...
Maybe.
Are there questions? I have a question. It might be a rather naive question, but if endpoints are so closely related, what is then the value of measuring different endpoints to calculate the BMD? So would you be comfortable to just take one endpoint or analyze a study where only one endpoint has been measured? A hypothetical study I'm talking about.
Well, this is a little bit ahead, so we first need to more solidly validate or confirm this hypothesis. I have so far examined 5 or so studies. I think we would need more, and we should also maybe better understand what is going on from mode of action thought, maybe. So basically what this theory says is that when you apply a chemical to an animal or an organism, then at a certain dose there will start to be effects, and all those different endpoints respond at the same time. And then of course there are other endpoints that don't respond at all, apparently. And this sort of gives an idea of maybe we can think about a fingerprint of this particular chemical, saying, okay, those are the endpoints that respond, and in another chemical we have a slightly different situation, or maybe totally different situation where all the endpoints are different. And so that I think also from a biological standpoint, this opens new interesting questions.
Laura, please.
Yes, I was wondering about the biological interpretation. I mean, what...
Turn on the microphone.
Sorry, Laura Martino from EFSA. So my question is about the biological interpretation of the response when it comes to this transformation that you are proposing to express as a sort of fold of the maximum response about one endpoint, no?
Yeah.
So in that case, because the interpretation of the benchmark dose is quite straightforward, I mean, it is very intuitive and works very well also for the biologists, the medicians that have to use the results. In this case, how would you suggest to interpret these results?
So you're talking about the interpretation of the different benchmark responses?
No, I mean, because at the end you would end up with a unique dose that represent different responses for the different endpoint. But those responses are expressed as fold of the maximum that you found among the various endpoints.
Yes,
so
of the log of the maximum response, so that's different. But anyway, so the idea is that in my theory I'm saying a small effect can be defined as 1/8 of the log of m. And so saying that a maximum response is a huge response, half of that log m is a large response, half of that is intermediate, half of that is small. So that's how I define a small effect. And then you can say, okay, that would mean that in red blood cells, for instance, that would be 5%, which coincides with the EFSA default. And you can also show that this effect size coincides quite closely to the 1SD approach that EPA is using. So the biological interpretation is nothing more than saying, okay, we think this is a small effect and it is in line with current defaults.
Yes, But I have the feeling that you are forcing back the definition of what is a small effect based on the numbers, I mean, that you get.
Yes, well, that, of course, there's always something arbitrary in defining what is a small effect. But the funny thing is that I just calculated what would that mean in terms of current defaults, and then it happens to be very similar. So that is a sort of a calibration, you could say, but the more important thing is if you want to define a benchmark response of 5% for red blood cells as a reasonable value, then that implies that for neutrophiles or for micronuclear cells, you need higher values for the benchmark response to make them comparable. And the idea of the theory, well, maybe if I have one more minute, the idea of the theory actually is that I distinguish between the impact of a treatment and the expression of the treatment by the endpoint. And as an example, you might think of a brake in a car. You can give a particular impact on the brake pedal, but that will result in different retardations in different cars. So the impact of what you do with the pedal is translated by the brake mechanism in different effects, retardations. So that's a distinction. And I'm saying now, okay, we have an impact of the chemical on an endpoint, but the endpoint, one endpoint will express that impact in another way than another endpoint. So that is the philosophical or maybe the biological interpretation of that. But I think it's more interesting to see when we apply this theory, can we then understand why those particular endpoints respond for this chemical and not for another endpoint? And maybe it's very interesting to compare, let's say, the adverse effects in pharmaceuticals with the intended effect of the pharmaceutical. Could there be a difference in that case?
Okay.
So there's a lot of things you can think of in the biological sense, but well, I don't think I have enough time.
Thank you very much, Professor Slob.
Okay,
we really need to proceed. We proceed with Matthew Wheeler, who has a rather challenging task to talk before the lunch. So he needs all your support, of course. We support you for this completely. Matthew Wheeler will talk about other quantitative methods for combining multiple studies and endpoints. Matthew Wheeler graduated, has a master's in statistics from Miami University and a PhD in biostatistics in 2013 at the University of North Carolina. He works as a health statistician for the Center for Disease Control and Prevention. Please, Matthew.
Thank you. Didn't have my CV, I forgot to send it. So I'm wearing this tie to show you that I'm an expert. So a lot of things I do are with model averaging as well as high throughput toxicity screening. I'm working a lot now with trying to develop response surfaces to describe chemical structure to the actual dose response in a high throughput toxicity screening, which recently I've had a little bit of a success with. But here I'm talking about multiple data sources and multiple endpoints. I was given the fun task of dealing with something that there's actually little done on and little data, so I get to kind of play around a little bit and ask questions, what if we had this, and look forward to that. So risk assessment is going to be concerned with looking at a single critical effect using a single study. We find some critical effect and we find a critical study, and that's how we typically go forward. We don't usually deal with multiple studies. We don't deal with multiple endpoints. We find our critical effect and our critical study. And I personally, as a statistician, don't like throwing out data, but it also can be problematic in terms of risk assessment. So what happens when there are multiple studies and multiple effects? So I want to spur discussion rather than promote any one method, because the methods I used are all just kind of to show kind of what happens, but not necessarily support any one research paper or another. So potential benefits, I like to say more data, better estimate of the dose response curve. Correlated endpoints. The reality is if we have endpoints that are correlated, if you have two cancers that given a particular insult to the system are more probable, then I start to learn about both simultaneously. So I have a simultaneous learning. And then multiple species. Well, could we get some idea of interspecies variability if we start developing these models and look at these approaches of extrapolation, if there are actual differences within interspecies variability that we really need to worry about. So the problem is that once we move into this area, we have a lot of complicated modeling choices, and not everyone is going to always agree. I had the privilege of working on the 83rd JECFA, and all the toxicologists in the room rolled their eyes and got mad when the 3 statisticians started to disagree on what model, and we all had 3 different points. So I can appreciate that there's a lot of different ways to approach the problem. So we have a lot of different modeling choices. We have overdispersion. Different studies are going to have specific study variability, but between species, between studies, there's different feed mechanism, there are different lab technicians that are working, pathologists are going to be different when they sack the animal and look at. There's going to be variability of defining this throughout the entire stream. So again, we have how do we define a correct model? This is again more assumptions. Modeling is an assumption that is often overlooked in the actual process. We kind of just often take for granted that this model is true and we go forward. And then the other thing is we're using software to do this, usually BMDS or PROST, but there's no off-the-shelf software that we can just jump in and do one of these analyses. It requires typically someone who is a little bit more adept at developing software and developing the methods than just a standard risk assessor might be. So what data should be included? That's another thing that was talked about this morning. What are the inclusion-exclusion criteria? How does that introduce bias into my analysis? Different studying links. How does that get included in the model? You have some studies that are short-term, 28 days, some are 90 days, some are 2 years. How does one combine all of that data into one systematic analysis? That's a very difficult question. I'm going to kind of play with a toy example just to show you how difficult it really could be. And then right now, is it research, or is there a possibility for some potential regulatory aspect? Can we actually say we know enough to use one of these more advanced methods, or is it more in the research realm still? So I'm going to look at a bunch of case studies of different data sets that I could actually find that had both. Multiple data sets with multiple endpoints, multiple studies. And there's a dearth of information out there, so I've got what I've got. There's not a lot of studies doing this, and then there's added problems. So I want to find ways that we can push science forward with existing data, and I'm going to stress throughout this talk that we have the problem where we open up a study and we get summary statistics. And that's how we model everything, basically on sufficient statistics. It would be better to have all of the data from the study. And I think that's something that we might want to start pushing for. Can we get the actual individual animal data? You can go into an NTP study and with a lot of work get such data, but it becomes more difficult when different authors are publishing. People have, rightfully so, territorial rights to their data. They want to do as much as they can with it to publish. So there's a lot of different things that we have to think about. But I want to say our goal is, how do we make risk assessment science better? So case study 1, this was at the last JECFA I attended, 3-MCPD esters. Now, there in reality are 2 studies that we have. The dose, part per million, those are already transformed by the toxicologist, so those are lining up, and we have 2 different endpoints. And I'm going to try to show what happens when I model the most sensitive, critical study that's been used, and then when I start to combine data and see what happens. And this was actually some controversy over this risk assessment because we had a 20-fold difference from what our recommendation was and what EFSA's recommendation was. So there was a lot of controversy about this point of departure that we chose. So we go through this, and the nice thing I like about this one is it kind of validates what I did at JECFA, which makes me feel right. I have, live in a house with 4 women, so I never feel right very often. So this kind of validates myself, especially with my teenage daughters. I'm right 0% of the time. So there's debate. EFSA and use the Cho et al. Data establishing, I believe, a point of departure of 4 parts per million. The JECFA, there should be a C, recommended 83 parts per million. The difference is based on constraining and unconstraining models, which can be a very contentious issue. But I'm going to avoid the problem. I'm going to use unconstrained models, and I'm going to look at all the data. And I'm only going to look at the log logistic model. There's a lot of dichotomous models out there. I'm only going to look at one model to kind of start to think about this. So if I look at just the hyperplasia, you'll see that these 2 studies line up pretty well. The Cho et al. Study actually just has a little bit of difference that causes a problem. You'll notice that the Cho et al. Study is the dark blue. And you'll notice, let's see if I can get this to work. Hey, that it's just a little bit lower in the background. And then the next high is just a little bit higher. It's not outside of this range, but that right there causes problems. If you unconstrain a model, what will happen is you'll get a model that goes up and across, especially when computing the BMDL, the lower bound. And then you will get a lower bound on that estimate, that point of departure that's much lower. So it results in a big difference. You can then take this model and say, well, okay, now what happens if I use some correlation mechanism between these endpoints and I model an average dose response curves between these? Fortunately, they're the same strain of rat, so I'm just using a study-specific variability. And I had to make a lot of assumptions in the background, Bayesian assumptions on the random effects and everything to make this work, because I only have 2 studies. But what we can start to see is that, well, I'm not modeling the second endpoint very well as an average, but there's a lot of heterogeneity in that second endpoint, and it's not even clear as what that means. So do we get a better estimate of hyperplasia? It looks about the same in terms of the dose response curve. So let's look at the brass tacks in terms of the benchmark dose lower bounds. How does this affect our estimate? Okay, if I just look using BMDS, the critical study, the single endpoint, my benchmark, and this is going to be unconstrained models. The BMD is 0.83, but as you can see, the BMDL goes 0.22. This was not used in the EFSA. I believe the gamma model went all the way to 0.04, and that was actually what was used in their assessment. But if you constrain the model, the BMDL is actually only 0.83. So once you constrain it, things change. Now I add both studies. Everything starts to line up. These are still unconstrained models. My BMD goes to 0.48. My BMDL looks at 0.96, which is pretty close to what the BMDL using the one study is. And things start to line up and make sense. But what's interesting is the uncertainty around my BMDU increases. Now it's 2.83. And then when I add even further a correlation between the endpoints, so now the 2 endpoints are correlated, and I start to talk about correlation, the BMD doesn't change very much in terms of the critical study, but the BMDU starts to go up. Now, this is just kind of an illustration. I had to make a lot of modeling assumptions to do this. There was, some sensitivity needs to be done with all the choices I made, but these were models using random effects to illustrate what could be done if I had a lot of different studies, multiple effects, and I start to combine those effects, and I start to look at correlation. I start to talk about this response surface that we were talking about earlier versus one individual point. And all BMDs were calculated using the same definition. So that's one study. So issues, limited number of studies. especially with long-term studies, there are very few long-term studies available. Everybody knows these are expensive. I can't necessarily learn the correlation coefficient, so I kind of fixed it from a Bayesian sense. I put some distribution over it of what a reasonable correlation was, but are the results robust? Do I do a sensitivity analysis? And could we sidestep some of these issues if we had the individual animal data? So what I mean by this is an individual animal, a pathologist will go down and look at the different types of adverse events that are under the study. So we can start to see that when event A happens, event B also typically happens. And those are things that we could then use to model if we had the data. So let's go forward. This is multiple study links, different links. Multiple studies, different links. So there's different links which the rats were exposed to. To do this, I found in NTP studies that have the same chemical. There's a lot of studies where they do with the same chemical, but they have different links, multiple endpoints. These are all the subchronic studies. And how do I integrate the information? Now I'm not going to talk necessarily about, well, I am going to talk about estimating risk, but I'm not going to talk about the benchmark dose specifically. What I'm going to just talk about is modeling this dose response curve even before we get to the benchmark dose. What happens? And what does a BMD mean in this context? If we're talking about time to dose, how do I talk about that? There's a specific amount of time needed to have a dose. So there could be a high dose but a short time frame and you're okay. But it could be a lower dose with a longer time frame and you have, so BMD is now related to time of exposure. So there's other things in play here. So, and I'm a firm believer that a risk assessment, if I'm doing it for the public, I need to use all information that I can use to be as objective as I possibly can. So I'm going to start off with, I think we have a rosy view of the world right now. We pick a single study, a critical endpoint. Now, this, and then I say, well, I have a certain amount of risk associated. So what I can say here is, think of these as tolerance distributions. As the dose increases, the probability of having some adverse response increases. So I have two adverse responses. Let's pick, say, two cancers, lung cancer and liver cancer, okay? And when I set a point of departure, I'm essentially setting it from the minimum point of departure, and that is going to be across all endpoints. But what is that really saying? Well, first of all, if there's two cancers and they're related, can I really say one cancer is worse than the other? I mean, how would you like to get cancer diagnosis and your oncologist say, you know, this isn't as bad as the other one, you're in good shape. You know, you're not going to like that. So we want to control risk for all cancers. But what are we really doing? Well, we're setting a limit, this gray line. And then we have another limit here that we set with this other one. Now, if I have a person that's negatively correlated, so these two cancers are negatively correlated, and this is a person's tolerance distribution, what I'm really saying when I specify the risk is that these guys are negatively correlated, and there's nobody down here. That is, there's nobody. The risk of getting lung cancer if you have liver cancer is very small, and vice versa. So that's how would I control risk. I'm basically saying that I'm really controlling at a 0.05 level, and I'm forgetting about these guys over here, even though it's less sensitive. Well, more realistic, we have some bivariate nature of the response. Is it, take two endpoints like lung cancer, is it reasonable to assume they're negatively correlated? And I don't think that's right. So this is more realistic. Let's flip this around. I now have the same point of departure for each study. And what would happen here, if I chose this particular point of departure, I'm going to now have people that have a risk of getting both. And these people are the ones that I'm predicting below here. And the reality is, if I chose, say, a 5%, this is based off 5% of the population, really my risk for that population getting all-cause cancer is about 7%. But this isn't the worst it can be, because in reality we have individual variability. If you look at diet, if you look at culture, if you look at race, genetic variability, variability starts to increase, and I have essentially one strain of inbred rat that I'm looking at, or a very specific thing I'm looking at. So the bivariate nature of the response is probably more reasonable, but is it reasonable to assume they come from a single population? So I would argue this is even more realistic. Now, you picked a study. Say the study was this one, but there's the entire population of rats. And you start to talk about your two endpoints. Now the probability that you are controlling for the cancers goes from about 5% to about 27% in this case. So the risk, once I start to talk about the variability of the entire population, goes up. And that's a problem when we just pick a single study. So we want to have some estimate of this variability. We want to really model as much as we can. The more data we have, the better. So we're a much more reasonable situation. Multiple subpopulations, positively correlated endpoints. Now the real problem is, how do I show you an example of this type of data set? So Jose pushed me, and I found a data set. It was hard. But I want to put something together and make something out of it. So I'm going to do what I did previously. I'm going to say there's random effects between studies. There's random effects between species. I'm going to use one model, the Hill model, because this is going to be continuous data. But now my next question is, well, these studies have different dosing mechanisms. They have different days under study, different things. How do I then add that component? We start to get more complicated as we think about this. So these are some NTP data sets that I chose. And to see it, look at this doesn't say the number of studies, but these are the number of doses and the links. You can see all those doses and then the links that they were exposed to the different doses. So now I have, in reality, some PBPK mechanism, but I might not necessarily have enough to describe that, so what do? We have all of this information. There's a lot to start to consider. You make the model more and more complicated, and the more complicated you make the model, some would argue, and I would argue that your reasoning starts to lay on a strand of, thread. One thing's wrong, everything falls apart. So we have conceptualized a model for internal concentration. So those animals had some internal concentration. They had some PBPK, I think would be most appropriate, but some simple scaling. What happens if I start to think about just simple scaling? I know this is done in the epidemiological literature, right? Often they use dose or concentration to some power. They'll use some sort of offset in terms of time. I want to think about this. So I'm going to look at a hypothetical model for acetaminophen for 3 continuous endpoints and see what happens when I model the data jointly and when I start to model not only the data jointly, but then under different definitions of dose. Because now dose is really dose times time. There's a time component there that I have to, unless I start to think about other things, I can't disentangle. So this is kind of what happens with 3 different responses if I scale it with time to the half dose. And this is the least responsive. And then these 2 are about the same. But you'll notice you have these weird data points over here. So the question is this just random variability, or was this a bad transformation? Should this actually be on scale dose further down? That's a question. So you do another one. Now the fit starts to change. You get more of a shape that looks more like Hill model in this. This starts to plateau really quick. You still get that, but you get a lot more moved down, so time to the 2/3 dose. But there's a lot of options, there's a lot to worry about, and what is benchmark dose? I didn't want to talk about it because you have that next thing as well. If this is my scale dose, how do these 2 things actually go back and forth? And since it was a theoretical exercise, I just wanted to throw that out there for people to talk about. How do we deal with dose? Okay, epi type thing, PBBK, but dose is now a function of time, and thus any BMD is essentially a function of time as well in most cases. So we've got to worry about that. So this last one was my pie in the sky. So we've got multiple studies. Now I'm talking about complete holistic integration of evidence. Now I'm saying that I'm interested in combining and looking at, say, in vitro studies relation to in vivo studies. Say I want to take a high throughput data set and relate that to some upstream endpoint. I tried to pull it off of something, it's very difficult. So I'm just going to say, well, the possibilities out there. I've been able to do certain things with these things. I can predict a dose response curve of an in vitro study a little bit with chemical structure, but you start to have other problems down the road when you start to link it the way. So develop a model that looks at correlation between a dichotomous endpoint with a known pathology. So the idea is, if you have a known pathology, you know how to deal with it, and then you want to look at other continuous endpoints that you not necessarily have an idea of how to deal with it. And what I did was I took all NTP short-term studies for all chemicals. You've got standard study to study variability, complicated, and what is the model form. These things all start to blow up. And the conceptual model is we have an endpoint we don't know what to do with. And it has some dose response. And we have a bunch of those endpoints. And then we have an endpoint for which quantitative risk assessment is routine. We know what to do with. The question is, can I build some correlation structure so in the future, if you just get these endpoints, can you predict some response curve over here? Can you get some estimate of what the benchmark dose would be so you can start to go back and forth between the two? And that was the hope. Unfortunately, with the data set I had, I drew blanks. There was nothing really there. But the endpoints that were less comfortable with, at least I myself am, I know Professor Slob likes continuous endpoints versus dichotomous, so it's different. Continuous endpoints, some of the meaning is not necessarily clear. In vitro assays, we have this explosion of high throughput data that I think in terms of putting it into a dose response, in terms of an actual risk assessment, there's a big question mark right now. And then genetic dose response. I don't think we have really any clue what to do with that data. We have some ideas, we have some correlations, but can we build a model that will better predict it using all of this data? And I think that that's somewhere where we need to start thinking and where we can go, but we need to have this data. We need to tie these two groups of studies together. So, conclusion of thoughts. Fortunately for me, there were many different ways to look at this problem. You can have the simple case where I have a risk assessment, I have the data that are the same, the dose is scaled the same, and I just have multiple studies. So the model is just slightly more complicated than what we're used to. Then you start to go down to the case where it's not clear how to scale the doses, and it's not clear of how to scale the correlation, and you make a more complicated model. And then you have the third case where it's not really clear what to do. And where do we start to decide this is research versus an actual product where we can say this is a risk assessment, we're confident in this. So I gave 3 possibilities, but there are more. I'm sure everyone out there can think of many more. So my big thing is, regardless of the problem, we need data. The hardest problem right now is not the statistics, but the fact that we're using methods that were developed in the 80s, dose response methods, because our data are so sparse. But there's a lot of data that you're generating, and we can take this data and we can develop a better understanding of the problem. But the summary statistics in your standard journal, they don't cut it. We have to use those older methods. We have to use something that's older. So the question is, well, if I'm a statistician, I need more data. There's much more advanced analytic techniques that can come into play. So I would like to argue raw data, the data needs to be available. I understand as scientists we're kind of territorial, especially if you spent all this money on your study and you want to get as much bang for the buck out of it as you can for your own publications, but at the same time, reproducibility comes into play. If a regulatory agency wants to look at the data, to be able to reproduce your analysis, and then the second part is some ways for posterity. In 10 years, there might be other methods that can take your data and do something with it that you didn't think was possible today. And then the other thing, and this is always the case, we need to talk about well-designed studies. I would argue that the standard study dose response now of 4 dose groups, the geometric scaling, is not necessarily the best way to go. From a statistical analysis standpoint, it would be just as ideal to have a random dose response across the range, but I know a technician is going to have a lot of problems having 200 animals and each giving them different doses. But I think we can argue that there's better ways to develop our studies. So I'd like to thank you. And
Thank you very much. Thank you very much. It was a fascinating talk. With the look into the future was really interesting. Are there burning questions? Matthias?
Yeah, thank you very much for this talk. Concerning your last recommendation about putting out raw data, which I think everybody could agree to, do we have already some ideas about the metadata that should go along with releasing such information, and what could be incentives for the researchers to release their data?
well, I think that that's a very good question, and we need to develop some international standards. I was talking with Jose and Marc on the phone last Friday about developing a metadata type approach. It would be nice to do standards-based. If you're going to do this type of study, this is the pathology that we would like to see, because often, some pathology isn't reported in some studies. Other studies report different pathologies. So you have that. And then I think in terms of an incentive, one might argue that a data repository with some level of citation, publication should be encouraged. I think that alone is a contribution to the scientific community. It's not just your study, but you have your data, and that's a contribution as well, and that should be rewarded. So those are my thoughts.
Dick Sime again. Well, thank you for your talk. I would like to add to this conversation that if you're talking about raw data, I think we usually mean here data on the animals and the duration and the type of effect and so on. But I would also like to make a plea for the chemical part of it. And certainly for in vitro test systems, chemicals do have a tendency to evaporate or to adhere to glass walls or to the wells. And that will have an effect on the actual dose. So the dose response also is, the dose part is very important to really pay attention to. Thank you.
Thank you.
Yes.
David Knoll, Matthew, have you thought of using studies from regulatory studies? Because you will have all the raw data in a good GLP study. And you should have, according to guidelines, a range of timescales.
I just actually got into this problem, depending upon the regulatory study, depends upon where the data are. So if I'm using an NTP study, it's regulatory, I can get into the NTP database, but the NTP database, to get into it, there's a very nice website that you can get all of the raw data. But from that point, you need to be a data scientist. In the background, I actually had a graduate student working on some issues with this to just take the raw data for the liver, and that's why I tried to do what I did with the third case study because I actually have that data. But it is, from the first question, we have to talk about harmonizing a way to get at the raw data because everybody has their raw data differently. So just to do this process requires weeks just to get it ready for an analysis, if not months to get the next thing. So there's a lot of steps, I think, but you're right, some of those, the raw data are available. And when they are available to go through that, that would be correct. So thank you.
Thank you very much. We will break for lunch now, but before we break for lunch, we expect you back in your respective breakout groups. So to keep it simple, the breakout groups are the floor above us. Breakout group 1 will be in meeting group 1 and will be chaired by Holger Schunemann. Holger, can you just wave? Holger? Breakout to GRU on bias-adjusted meta-analysis will be chaired by Sophia Diaz. Sophia there, which is a meeting room 2. Breakout group 3 will be chaired by Donald, who we all saw in the beginning, and will be in meeting room 3. And breakout group 4 will be in meeting room 4 and chaired by Marc Aerts. We see each other in the breakout groups at 2:30. Please, there will be a coffee break at 4:30. Please respect the time for the coffee break and take your nameplates to the breakout groups so that you are identifiable. Thank you very much and see you later.